The topic of my talk today is adaptive radiation. And when you think about the classic cases of adaptive radiation, the ones you see in the textbooks, they are almost all from islands. So of course, the Galapagos Darwin's finches, the Hawaiian honey creepers, the Hawaiian silver swords, even the East African Rift Lake cichlids, where the Rift Lakes are essentially aquatic islands in a terrestrial sea. And so in fact, I would say basically all or almost all the classic cases are from islands. And this phenomenon is so pervasive that some people have even suggested that adaptive radiation is an island phenomenon. Now, I don't necessarily agree with that viewpoint. I don't agree with it, but I certainly understand where these authors are coming from. So what is it about islands that fosters adaptive radiation? Well, the idea is something called ecological opportunity, that when many islands arise de novo in the sea due to volcanism or whatever, and initially they're empty, and then they're colonized by plants and animals and microbes, and a community builds up. But those species that arrive there find a depauperate fauna that compared to the mainland from which many of them come. And as a result, there are many niches that are empty, if you will, let me use that idea. And so there are opportunities for them to use resources that simply aren't available on the mainland because other types of species usurp those niches. And so the idea is that this ecological opportunity is the opening for adaptive radiation to occur. Well, this idea is certainly, it makes sense. It's plausible. It's also not easy to actually test. And one way that one might go about examining this idea is by doing an island to mainland comparison. Take sister taxa, the same sort of organisms, where one clade occurs on islands and the other in the mainland, and try to figure out what goes on, what the differences are, what allows one clade to evolve differently than the other. And so what I would like to do today is to provide an example of such an approach. And anyone who knows my research will not be surprised that that example will focus on lizards in the genus Anoles, the animals that I've spent my career studying. And here's just some of my favorite Anoles. So before getting going, let me just introduce you to Anoles. Anoles are characterized by two features. One is the presence of expanded toe pads. You can see them here on some of these lizards that give them a great clinging capability. These toe pads are convergent to the ones that have also evolved in geckos, with which many of you may be familiar. The other trait that defines anoles is the presence of a dewlap, an extensible throat fan that is found in all but two species of anoles. It's in those species, males always have a dewlap. Females are variable in whether they have a dewlap or not across species and in the development of that dewlap. Now, you all in North Carolina are very fortunate in that you live in a place where anoles actually occur. And so you may have seen this. Many male anoles spend much of their life doing this, just sitting there and sticking their dewlap out time and time again all day long, basically saying, if you're a male, stay away, this is my territory. But if you're a female, come here, I'd like to get to know you better. In addition, they also dewlap towards predators. So the dewlap is used in many communicative contexts. I just want to point out that the diversity of dewlaps across species is spectacular and a topic of great interest to many people. So I want to start by talking about research on anoles in the Caribbean, where I have done much of my work throughout my career and where the vast majority of research on anoles has occurred, and particularly on the islands of the Greater Antilles, and that is Cuba, Hispaniola, Puerto Rico, and Jamaica. And on those islands, there are two patterns of interest in the anoles. First, on any island, if you look at the community of species that occur together, you will find species that differ in their morphology, ecology, and behavior. So for example, if you go to Puerto Rico and go to the rainforest in the Luquillo Mountains, and if you enter the rainforest and then stand quietly, after a few minutes, the lizards will forget you're there and they'll become active, and you'll realize that there are species all over the place. But each species is in its own part of the environment, and the species differ in their appearance and in their behavior. And the differences in anatomy seem to make sense, that species on narrow surfaces have very short legs that they use to move carefully as they negotiate those narrow surfaces. Species near the ground have long legs and they run and jump and so on. Now, the most interesting thing about these communities of anoles is that you see the same set of habitat specialists on each island. So for example, I mentioned this one that uses narrow surfaces, what we call a twig anole. If you go to, say, Jamaica or Cuba, you will see a species that looks essentially the same, living on the same habitat and behaving in more or less the same way. And for the most part, this is true for each one of these different types of habitat specialists. Now, I need to give you a piece of jargon we use. We call these habitat specialists ecomorphs, and they're named for the part of the environment where you normally see them. I want to briefly introduce you to the Greater Antillean ecomorphs. Trunk ground anoles, as their name implies, are found usually on tree trunks down to the ground. They're characterized by having very long hind limbs. They're stocky, muscular animals. Their toe pads are not well developed, and they're generally brown in color. And here are the trunk ground anoles of several islands. The trunk crown anoles are also found on tree trunks, but they're found basically at eye level up to the top of the trees and into the canopy. They have shorter limbs than trunk ground anoles, a longer head. They have very well-developed toe pads, and they can turn colors from green to brown. And here are the trunk crown anoles of the 4 islands. Next are the crown giants. As their name implies, these are the giants of the anole world. They're bigger than I can show you on this little screen. They get to maybe 1.5 feet in length. They have a large head, a vertebral crest. Most of them can change color from green to brown. These are also the most aggressive of the anoles. They will stand their ground and defend themselves, and they do have very sharp teeth, in case you're wondering. Here are the crown giants of several islands. Next are the grass bush anoles. These are small lizards that live on low-lying, narrow vegetation, actually on grass stems, or as you can see here, ferns or bushes. They have short forelimbs, they're very slender, a long head, and their most interesting feature is that they have an extremely long tail, sometimes as much as 4 times the length of their body. And we're still not sure really what that's all about. Here are the grass bush anoles. Finally, the most extreme of the ecomorphs in really all respects are the ones I've mentioned already, the twig anoles. They use very narrow surfaces where they creep very cautiously through the environment. They have very short limbs and tails, a very long head, and they have a light camouflaged color. And here are the twig anoles of the 4 islands. Well, the first question that you might ask is, how is it that you have these same habitat specialists on each island? What is the evolutionary story? One possibility is that each habitat specialist type has evolved a single time. And if that were the case, say all the twig anoles, although occurring on different islands, would form a clade, a monophyletic group. And that would imply that twig anoles evolved one time and then somehow ended up on 4 different islands and became different species. And it would imply similarly that the other ecomorphs also had evolved a single time. The other possibility, of course, is that each one of these types has evolved independently on each island. And that is, this latter possibility is in fact what happened. In this slide now, the colors represent the different islands. You can see the trunk crown anoles on the 4 different islands are not at all closely related. And this is true essentially for all the different ecomorph types. Here are the trunk ground anoles, 4 separate clades, the twig anoles, the grass bush anoles. So the anole phenomenon, the ecomorph phenomenon, is a great example of convergent evolution. And more than just convergence, it is convergence of entire communities, entire assemblages of habitat specialists. This is an idea that there are more, this what we call replicate adaptive radiations. There are more and more examples being proposed in the literature every year, but I would say that the anoles are probably still the best documented case of this. Now, when we see convergent evolution of species occupying similar environments, that is strong suggestive evidence of the role of natural selection, that it can't be a coincidence, for example, that species up in the canopy have independently evolved to be green time and time again. It just seems unlikely to occur by chance. Rather, it suggests that being green is adaptive for living up in the canopy. And in this case, the reason, of course, is obvious, for camouflage. Well, work on the adaptive basis of the variation among ecomorphs has focused on 2 traits primarily. That is the toe pads and how long their legs are. So let's start with the toe pads. This is a brown anole, a trunk ground anole, hanging from a microscope slide. I hope you can see up here, it's a microscope slide. It is hanging on by a single toe pad. And remember, trunk ground anoles do not have well-developed toe pads, and yet it can hold its own just by 1 toe. We now know how this works. The toe pads are covered with millions of microscopic little hairs called setae, and adhesion results from intermolecular bonds. The electrons on the setae forming bonds with the electrons on the surface, something called van der Waals forces. And that is how they're able to cling to smooth surfaces. And again, as I mentioned, geckos have evolved a very similar system independently. Well, more to the point with regard to anoles, the topads vary among species in a way related to the environment. So for example, more terrestrial species like this grass bush anole have narrow topads compared to the more arboreal species, and there are also fewer scales making up the topads, what are called lamellae. If you counted them up, there are many more scales here than on this one. And when we look across ecomorphs, what we find is that the number of lamellae on the topad is greatest in the two arboreal ecomorphs, the crown giants and the trunk crown anoles, and it's lower in the more terrestrial ones. The same is true for topad area, but I'm not going to show you that. Now, just a shout out to some very cool work related, not the topads, but the claws. Recently, some workers at UC Berkeley have shown that there are differences in the ecomorphs in claw size and shape as well, another aspect of hanging on to surfaces. We tried to do this years ago and were unable to, so I was delighted to see this paper. The other main feature that we focused on is how long the legs are. These are silhouettes of actual museum specimens. We put them on a Xerox machine, if anyone knows what that is anymore. And so these are real silhouettes of lizards. It shows the difference in the body size, incidentally. This is a crown giant and this is a twig anole. But if you can mentally subtract body size and look at the length of the legs relative to body size, I think what you can see is that the twig anole has very short hind legs relative to its size, whereas at the other extreme, the trunk ground anoles hind legs are much longer. So there's great variation in relative limb length, and that too is related to the environment. You can see a very strong relationship between forelimb length and the perch diameter that a species uses. The relationship with hind limb length is messier, but still statistically quite significant. So in studies of this, of ecomorphology, many researchers get to this stage, they find a correlation between morphology and ecology, and then they're kind of done. They say, well, the reason that you see green color up in the canopy is it's camouflage, or the reason you have bigger toe pads up in the canopy is to cling better. That's certainly, these explanations are usually very plausible, but they're really more hypotheses to be tested rather than explanations per se. And in particular, one approach to try to understand such correlations is to look at what we call performance capability or functional abilities, and basically ask, what do differences in morphology do for the organism? What difference does it make how long its legs are or how big its toe pads? And in turn, how do those different capabilities relate to where you find them in the environment and what they're doing? And so we've taken this approach with anoles, and we focus on three aspects of measures of performance that are ecologically relevant: how fast they can run, how far they can jump, and how well they can cling. And in terms of how fast they can run, I'm just going to show you this cool video so you can get a sense of what a lizard running is like. But obviously, running ability could be relevant to capturing prey and eluding predators. And look how it uses its hind legs. I'll come back to that in a moment. Similarly, jumping ability is important for the same reasons. And finally, clinging ability, which we measure on a force plate, it determines how well they can hang on to surfaces. Well, those are the measures of performance that we have done through the years. And I'm not really going to go into any detail, but the predictions one would make are pretty straightforward. Species with longer legs relative to their body size should be able to run faster because they can take longer strides, like in that video. Similarly, they ought to be able to jump further because they accelerate in the jump through a longer time and so can achieve a higher jump velocity. And lizards with bigger toe pads should have greater clinging ability. Well, without going into any details at all, I'd be happy to talk about this later. These predictions turn out to mostly be correct, with some interesting twists that we didn't expect. But for the most part, we felt, based on this work on Greater Antillean anoles, that we had a good understanding between the relationship between morphology and performance. And so we could understand how these ecomorphs, differing in their morphology, how that affected what they could do. In turn, field studies on studying what they do illustrated why these differences in performance were relevant. So we thought we had a good idea of the adaptive basis of anole evolution in the Antilles. And as I said, I just don't have time to review those studies, but I'd be happy to talk about it more later. With that, that was my whirlwind tour of Greater Antillean anole evolution. And I now want to shift to what I could call the dirty little secret of anole biology. And that is, those of you who don't know lizards or herpetology, but have read papers on anoles, I could understand if you thought that anoles occur in North Carolina and the Caribbean and nowhere else, because the vast body of the literature on anoles is on the islands. But in fact, anoles are found throughout the neotropics. And indeed, there are more mainland species than there are in the islands. And let me just show you a couple of mainland species, because a lot of them are really cool. And in fact, I want to give you two really cool mainland anole facts that are completely irrelevant to this talk, but I think they're worth telling you about. The first is that many mainland anoles have ginormous dewlaps. Take a look at some of these species. They're huge. We have absolutely no idea why they have such large dewlaps and what they do with them, but it is fascinating. Secondly, females of many mainland anoles have large dewlaps, as large as the males do. This is very unusual in the Caribbean. And sometimes the dewlaps the females have are different colors and patterns than males. Here are 5 species with the male on the left and the female on the right. You can see different colors, different patterns. And this is not unusual in mainland anoles, very unusual in the Caribbean. Again, no idea why this is. Great, one of many great anole projects waiting to be done. But that's not what I'm going to talk about. About a dozen years ago, I wrote a book on what we knew about anoles back then. If you had looked at that book, you would have seen that I don't talk about mainland anoles all that much. And the reason is that the vast majority of the research, particularly the ecological evolutionary research on anoles, has been done on Caribbean species, even though, as I pointed out, there are more species on the mainland. So how do we explain this discrepancy? Well, I think there are 2 explanations. One is anoles on the islands are just so much more abundant and active on the mainland. If you want, it's not an exaggeration to say that in some places on the islands, if you go out to study them, the rate limiting step is how quickly you can record the data. They're just everywhere. And so collecting data is quite simple, all kinds of aspects of their natural history. By contrast, in the mainland, anoles can be hard to find. At the extreme, if you're in the Amazon, a good day is when you see 3 anoles, whereas in the Caribbean, a good 5 minutes is when you see 3 anoles. And so it's just so much easier to collect data. And for people who start working in the islands, it's just too much trouble to work in the mainland. So undoubtedly, the ease of study is one reason that the island anoles have been studied so much more intensively. The other reason, though, is the history of anole research. And that goes back almost a century now to someone named Thomas Barber, who was the director of the Museum of Comparative Zoology at Harvard. And as I think these photos illustrate, he was quite a character. He was also quite wealthy. He owned his own yacht, and he would go on these long expeditions sailing through the Caribbean on general collecting trips. And if you're collecting herps in the Caribbean, that means you're collecting anoles. And so he amassed an enormous collection of anoles at the MCZ. Well, his successor, I think 2 down the line in herpetology, was a man named Ernest Williams. Williams did his PhD on fossil turtles, but when he arrived at Harvard, he saw all the anoles in the jars and made that his career's work. And basically, just about everything I'm going to talk about today traces back in some way to the foundations laid by Williams. One of the things that he did was he trained an extraordinary group of graduate students. Pictured here is Stan Rand, who some of you probably knew, who worked in Panama. He was Williams's first student. In fact, Williams trained an extraordinary group of people who went on to be leaders in evolution and ecology. And as I said, most of the work on Caribbean ecology, at least until recently, there's been a real blossoming of research in the last decade or two. But before that, most of the work could trace its way back to Williams one way or the other. And so to a large extent, that also has been why, the point I forgot is Williams worked in the Caribbean, following up on Barber's collecting. And so that also explains why so much work has been done in the Caribbean. I do want to acknowledge that there has been some work in the mainland. They're not completely unstudied. And one person more than anyone else who's been responsible for shepherding that work is Jay Savage, who had a fabulous lab at the University of Miami. And he and his students studied the herpetofauna throughout particularly Central America, very extensively. All right, so I am now going to talk about mainland anoles and what we know about them, and what we know compared to the islands. And I should tell you, this is pretty much a new talk for me. I gave parts of it about a year ago, but it's mostly a new talk. And you might ask, so what's new? Why am I talking about mainland anoles now? There are two reasons. One is we finally have a phylogeny for mainland anoles. It was published a couple of years by Steve Poe and co-authors. It is an all Anoles species phylogeny, and it's a good start. We've got more work to do, better data are coming along, but it gives us a phylogeny to work with, which before that we didn't have. Secondly, there was a very large project that went on for about 10 years that I was involved with, where we collected a ton of data on mainland anoles, data comparable to what had been collected on island anoles for many, many years. I need to recognize Anthony Harel of the Natural History Museum in Paris, who really led much of this research. And here's just the sort of data we collected. Morphometric data on more than 200 species, performance data on 131 species, habitat use data on almost 100 species, and even behavior data, filming the lizards and seeing what they do for 60 species. So it's a great set of data, and it allows us now to ask questions about how the mainland and the island radiations of anoles differ, and to compare the patterns of evolution. So let's get going. We'll start with the phylogeny. Anole phylogeny can basically be broken down into three important groups. The first group are the mainland group, sometimes called the dactylo anoles. These are the sister taxa to the rest of the anoles. There are 83 species. Not all of them were included in this phylogeny. Now, I do need, well, all right, there's the Dactyloa group, sister to the rest. They occur on the mainland. Very cleverly, we called them M1, mainland. Sister to that, then we look at the remaining clade, and we see this large group of island anoles here, 140 species. That group, however, is not monophyletic because a second mainland group arose from within the island radiation. This is the Norops clade, or M2, because they occur back in the mainland. And as you can see, this M2 clade actually has the most species compared to the others. It has more species than the older mainland clade. So putting a biogeographic spin on this, one thing I should point out, the sister taxon to anoles is almost surely a mainland species. We're not actually positive what the sister taxon is, but the candidates occur on the mainland. So what that means is that anoles arose on the mainland. We have our basal clade here in the mainland. They probably arose 50 million-ish years ago. It's a very old group. Then the first thing that happened is 43 million years ago, they colonized the islands. I should point out that this mainland group is almost exclusively in South America. So presumably anoles arose in South America. Then from South America, they colonized the islands and basically diversified all of the island anoles, with one little exception I'll get to in a minute. All the island anoles are the descendants of this one colonization from the mainland. Then 10 million years later, about 34 million years ago, a species colonized back from the islands back to the mainland. And there are two arrows here because we're not sure where that colonization occurred, but it was somewhere in Central America. And so that's the basic story of anoles. The one side point is there is one little clade here coming out of a sister to M1 that colonized the southern Lesser Antilles, so a second island radiation, but it only covered the islands from basically Grenada up to Martinique, and there are only 8 islands there, and so it's a very small group of species, not important for our story. So the basic point is 3 major clades: the ancestral mainland clade in South America, the clade that colonized and diversified and all the adaptive radiation I told you about, and then one species that came back to Central America and diversified widely on this return colonization. And you can see this return colonizer actually almost twice as many species as the older mainland clade, an interesting discrepancy in its own right. And we'll come back to that. All right, so here are the questions I want to talk about for the rest of the talk. How does our adaptive radiation differ between island and mainland anoles? That will be the big question. But secondly, focusing on that clade that returned to the mainland from the islands, what is it like as it radiated? Is it like the other mainland clade? Did they basically convergently evolve their adaptive radiations? Or does this clade maintain a similarity to the island clade from which it diverged? So in other words, is the second mainland clade more similar to the first clade or its island ancestors? And finally, what I think is the most novel and interesting question is, you have these 2 mainland clades. one in South America, one in Central America. Eventually, they came together when the Isthmus of Panama emerged. What happened when these two clades come together? So let's start with the first question, just comparing island and mainland radiation. We can ask a number of different questions about how these two radiations, how they differ. This is simply a look at the position of species in morphospace, a principal component analysis on size-adjusted data. And two patterns should jump out to you. I should point out that the island species are filled circles, the mainland species are open symbols. What you see first is that the mainland anoles occupy roughly the same morphospace as the island anoles, maybe a little smaller, but roughly the same. And so immediately, we have to recognize that the island anoles are not more extensive in their radiation than mainland anoles. They're roughly comparable in their morphological variation. Secondly, although they are overlapping greatly, they do not do so entirely. There are some differences. In particular, the island anoles have radiated in this space here that the mainland anoles have not touched. And there is a smaller space down here that the mainland anoles have diversified in and not the island anoles. So they're not perfectly congruent in terms of their radiations. Now, when we look at habitat use, using the two aspects of habitat use that are commonly measured in anoles, we see that if anything, the mainland anoles are more diverse ecologically. They occupy ecospace here that the mainland, that the island anoles do not occupy. And if you also looked at, this is of course microhabitats, if you looked at macrohabitats, mainland anoles also are found in places that you don't find island anoles. For example, up in cloud forests or on beaches. People think when I go to the Caribbean, I'm spending time on the beach, but why would I do that? The anoles don't live on the beach in the Caribbean, yet in the mainland they do. So in all respects, they're ecologically actually more diverse in the mainland. So one immediate conclusion, adaptive radiation is not an island phenomenon in anoles. All right, so a first question that may come to mind, I talked about the Greater Antillean or West Indian ecomorphs that have evolved on the 4 major islands. Do those ecomorphs occur on the mainland? And the answer is yes in a few cases. For example, here's a Jamaican twig anole, and here's a couple of mainland twig anoles, morphologically very similar, occurring on very similar narrow surfaces. And here's some more twig anoles, and I have to tell you, this is from a paper that was one of the most exciting papers to me, well, one of the greatest honors I ever received, because when I read this paper, I discovered that this handsome fellow here was named after me. It was the biggest thrill I've ever gotten. And it was really great because the people who did this didn't tell me about it. They just sent me the PDF of the published paper and said, think you might want to take a look at this. And so you got to admit, that guy is lovely looking. I have to go down to Southern Ecuador to meet my kin. Anyway, twig anoles definitely have evolved on the mainland. There's also a grass bush anole. Here's one from Puerto Rico. And there's a grass bush anole in Panama that's a dead ringer for the Greater Antillean grass bush anoles. There are also some crown giants, for example, Anoles fraseri. So there are a few examples of mainland species that conform to the West Indian ecomorph categories. However, most species do not. This is an old paper published by Duncan Urshik in the late 1990s. And it's, again, a look at position in morphospace. This is on, again, size-adjusted data, and we've graphed PC1 versus PC2, 3, and 4. The polygons are the morphospace occupied by the ecomorphs, and little circles are mainland species. And what you can see is that most of the little circles do not fall within the polygons. In other words, the mainland species are occupying morphospace not occupied by the ecomorphs. They are not ecomorph, they are not congruent with the ecomorphs. And in fact, if you go to the mainland and look at anoles, you will see this clearly. For example, these are 4 of the common species at the famous La Selva Biological Station in Costa Rica, and none of them matches with the ecomorphs in the Greater Antilles. There are a number of mainland species that are leaf litter specialists. That wouldn't seem to be a big deal, but the ecomorphs are not leaf litter specialists. They're always on the trees or other vegetation. Here's the beach anole I mentioned before in Venezuela. Again, a type of habitat not used by West Indian anoles. So most mainland species have gone a different way in terms of their adaptation to the environment. Now you might then ask, well, maybe there is a different mainland ecomorph syndrome, a different set of ecomorphs that has evolved multiple times across the mainland. And there is one clear example where that's the case, and that is what we call semi-aquatic anoles. These are species that are always found in proximity to streams. And by proximity, I mean within a couple of meters. And if you scare them, they will actually jump into the water and swim away from you. And they will forage at the edge of the water and possibly even in the water, unclear on that. And these species are very similar in their very robust, stocky morphology, very long legs. Yet the phylogeny makes clear that these semi-aquatic anoles have evolved 3 or 4 or possibly even more times. So that is one example of a convergent habitat specialist type in the mainland. We actually aren't sure more generally. This is a topic we need to delve into our data further to see. So I just can't give you an answer more generally, but at least there is this one convergent habitat specialist type in the mainland. I do want to add that the aquatic anoles, semi-aquatic anoles, have been in the news the last couple of years. A really cool finding that something about their skin keeps a layer of air around their skin when they're underwater, and they have evolved to be able to rebreathe the air that clings to their skin. This was first reported by Lindsay Swerk a few years ago, and then Luke Mahler at Toronto has shown that this has evolved actually convergently in a number of these different species. So very cool aspect of their biology. Okay, how about more generally, is the relationship between morphology and ecology the same in the mainland in the West Indies? Even if the ecomorphs don't occur there, is the relationship the same? And the answer to that actually is clearly no. This is again that paper by Duncan Urshik on a limited number of species showed very different ecomorphological correlations. And we've elaborated on this with our bigger data set and found very much the same pattern. I want to give you one example. It turns out that Caribbean anoles have bigger toe pads than mainland anoles. And here's just some work that Hannah Frank, who's now at Tulane, and Luke Mahler did, showing that at a given size, Caribbean anoles have larger toe pads. And in fact, if you look at the relationship between perch diameter and toe pad area, again, we see the Caribbean anoles for a given perch diameter, the Caribbean anoles have bigger toe pads. And so in a case like this, where we see a different ecomorphological relationship, there are two possible explanations. One explanation is that the relationship between morphology and performance is different. Now, in this case, what that would suggest is that perhaps Caribbean anoles just have crummy toe pads, that per unit area, they don't have as much adhesive ability. So to produce the same amount of functional performance, Caribbean anoles would need bigger toe pads. And so that's one possibility. Now I have to say, both in this case and more generally, that seems unlikely. We have a pretty good understanding about anoles functional biology. It seems like you would think a toe pad is a toe pad. Unlikely the Caribbean anoles have poor quality toe pads. Much more likely is the other explanation, that there are different demands in the environment, that for living in a particular place, greater performance capability is needed. That in this case, for some reason, Caribbean anoles need to be able to hang on better, and that's why they have larger toe pads. And that to my mind is the much more likely explanation for these different ecomorphological relationships. Well, I was sent this photo a while ago, which is really kind of a cool photo. What we see is these are two island anoles fighting with each other, and as they often do, and one male is biting the head of the other and is holding up both of their bodies, hanging on by its hind legs. Cool photo. Well, then I found other examples of the exact same thing. And it occurred to me, maybe this is one example of how Caribbean anoles need greater clinging capability. We know that Caribbean anoles fight with each other more, they're more territorial, more aggressive. And so maybe this happens regularly enough that there is selection for greater clinging capability. And so that might be the explanation in this case for why greater clinging capability, why we have a different ecomorphological relationship. And as I suggested, this agrees with my suspicion that it's the performance to ecology link that is different, not the morphology to performance link. Having said that, it turns out that in some cases I was wrong. And in fact, the morphology to performance relationship is more complex than we realized. For example, we looked at the relationship between hind limb length and sprint speed. Our research had shown that longer legged species run faster. We thought we understood that. Well, it turns out that for a given hind limb length, Caribbean anoles run faster than mainland anoles. So there is something more than how long your legs are that determine how fast you run if you're a lizard. Presumably that has something to do with the musculature, and we're looking into that now, the size of the muscles, the composition, or something like that that differs between island and mainland anoles. Another aspect of morphology I haven't talked about, but it's the determinants of bite force, and head height has long been thought to be an important proxy for that, but we find for a given head height that mainland anoles can bite harder than Caribbean anoles. So again, the skeletal structure isn't enough. There's something else. And again, it's probably something to do with the muscular system. And so we're looking into that as well. So what we found more generally is more complicated than we thought, that we need more detailed study of the biomechanics of these traits to understand differences in the morphology to ecology relationship. Having said that, my bet is that most of the differences have to do with the demands of the different environments and not the biomechanics, but that is still something that we're working on. All right, so the bigger question, how can we account for the differences in the patterns of evolution between mainland and island anoles? We see these differences in a number of different respects, the lack of ecomorphs, the different ecomorphological relationships. Why has that happened? Now, in addressing that question, I went back to an old paper by Robin Andrews from 1979, where she reviewed the data on the population biology of anoles. And what she found was that, this is in 1979, that mainland anoles, relative to West Indian species, mainland anoles have lower population densities, they don't live as long, they eat more food, and they spend less time foraging. They eat better food, bigger, better food, and they grow faster. So a series of 6 differences in the biology of these species. Now, 40 plus years later, these differences still hold up. And the reason they still hold up is that people have stopped collecting this sort of data. We desperately need more data on the basics of population biology to understand these differences. But assuming that Andrews's summary is correct, let's explore why that might be. Well, one possibility is that there are more competitors on the mainland, and surely that is true. There are many more types of insectivorous lizards, certainly a lot more insectivorous birds, even diverse frogs and other sorts of insectivores. So it could well be that mainland anoles are competing with a broad range of other species for insects. However, that explanation would not explain the trends that Andrews reported, because if competition is a problem, you would expect anoles to be eating less food, to be having to forage more, to be eating low quality food, and to growing more slowly. All predictions contrary to what the data show. So increased competition doesn't seem to be the explanation. Another possibility is greater predation risk on the mainland. Now, greater predation risk could explain these patterns. If there's greater predation, that would lead to lower survivorship, which would lead to lower densities of the anoles. And in turn, that would release species from intraspecific competition, allowing them to eat more food and eat better food and grow faster and so on. So it's very plausible that differences in predation between the mainlands and the islands might be the reason we see these different patterns. Now, are there more predators on anoles in the mainland? Absolutely. It turns out that to some extent, anoles are the snack box of the jungle in the mainland. Everyone eats them. For example, this is a quetzal. Quetzals are fruit-eating birds. And yet, what's that in his mouth? Not a fruit, it's an anole. There are many types of snakes that specialize on lizards, particularly anoles. Even things like this horrible monkey will eat anoles when they get a chance. So anoles on the mainland are preyed upon by a wide variety of different species. And that's important because that means there's not one anti-predator approach that can work because there's so many different types of predators. So very plausibly, greater predation risk on the mainland has led to different patterns of ecomorphological evolution. How might that work? Well, on the mainland, we would expect anoles must be much warier, much more on the lookout. Whereas in the Caribbean, it's a party all the time. They're running around, they're displaying each other, they're courting females. And so they just have very different lifestyles. And in fact, our data back this up. We found that island species are more active, they move more. They also flee at a greater distance from an approaching predator, and they flee greater distances. You might say, well, anyway, the reason they do that is mainland anoles just hunker down. If you walk up to a mainland anole, it just sits there and hopes you don't see him. And so you can get much closer. And mainland anoles display more. And so what I would say is, what I like to way to put this is you could have 2 anoles living on the same surface, the same structural habitat, one in the islands and one in the mainlands, and they have very different forces affecting their lives and thus must adapt in very different ways. And that would be the suggestion, I would say, to explain why we see these ecomorphological differences. Now, actually testing the role of predation as an evolutionary force, particularly a macroevolutionary force, is not easy. And I'd be happy to talk in the questions about how we might go about doing that. I do now, however, want to move to the second major question, and that is, how has the M2 clade, the one that came back from the islands to the mainland, has it radiated in a way more similar to its ancestors, the island form, or to the other mainland clade? So let's start by going back to this plot of morphospace that I showed earlier, showing that the mainland and island anoles are pretty congruent in their position, though not entirely overlapping. But let's focus on the mainland anoles. And here's what we see are the two clades of mainland anoles, M1 and M2. And you can see they are pretty much in the same morphological space. And what that means is that the M2 clade came from the islands and diversified in pretty much the same way as the M1 clade, that you could call it convergent radiations. Now, I realize a sample size of 2 is not great for statistical testing, but they have seemed, in terms of their morphological variety, to done pretty much the same thing, even though the M2 clade had island ancestors. Now, studies of evolutionary diversification and adaptive radiation posit that something called an early burst occurs, where a species gets into an area with ecological opportunity, niches are available, if you will, so they have a rapid burst in phenotypic evolution as they diversify to use different habitats, and a rapid burst in species diversification, and then through time, it slows down as the niches get full. This is an idea that has a moderate amount of support, although a lot of variation in the outcome in different clades. And so we looked at the 3 clades we're talking about, and I am calling the island group a clade, even though technically it's not because it's paraphyletic with M2. We looked at the 3 groups to see if there's evidence for early burst evolution in both morphology and species diversification. And this is work that was published recently in PNAS. It was led by Austin Patton, who was just finishing up his doctoral work. He's now a postdoc at Berkeley, and he really deserves the lion's share of credit for the really sophisticated analyses in these papers. He was working with Luke Harmon primarily, who is also a major force, and a number of other co-authors as well. So let's look first at morphological evolution. And using BAM, we looked at the rate of morphological evolution across the tree. And what we see is that the Greater Antilles, this group here, more or less shows that early burst pattern where rates of evolution were high deep in the tree. The redder the color, the higher the rate of evolution. And then at least in many clades, it cooled off through time and became much bluer. So more or less congruent with early burst in phenotypic evolution. The M1 clade shows it to a lesser extent. It's certainly much warmer here for the first 10 million years or so, and then cools off. So you get early burst more or less in the Greater Antilles and the M1 clade, but not in the M2 clade. The M2 clade starts right here, and it's barely colorful at all. And so there's really not much evidence for enhanced rate of evolution deep in the tree in M2. So there seems to be a difference between M1 and the Greater Antilles and M2. And the important point here is that the 2 mainland clades do not show the same pattern. Now you might ask though, given that the M1 and M2 are equally disparate in their morphological variation, how did M2 accomplish that if it didn't have this heightened rate of evolution? Well, the M2 clade had a different trick. They show an early burst in evolution in terms of species diversification. And so if we look at this diagram here, here's M2 showing very high rates of species diversification early on and then cooling off. And we in fact see this both in the HISI analysis and the BAM analysis. I do need to tell you that analyses of species diversification, as you probably know, are a real problem these days. There's a real debate about which of these methods, BAM or HISI, is the most appropriate. And then recently, Luca and Pennell argue that none of them is appropriate, that they're fatally flawed in trying to reconstruct these histories. They argue that the best you can do is calculate an overall rate of diversification with the assumption that speciation is constant. And in their analysis as well, M2 early on has a high rate of speciation. it's really complicated. I've already spent too much time on it. The short story is the evidence consistently, all three methods shows an early burst in the M2 clade and does not show an early burst in the other ones across all three analyses. Not a particularly satisfying answer, but there does seem to be a difference as much as we can interpret this. Another thing we could look at is the extent of convergent evolution within each of these regions. Now, and so basically this graph shows the most extreme examples of convergence connecting a species on one side to another species on another. That's just to document the convergence. A lot of convergence in the Greater Antilles, as we would expect. A lot of convergence in the M2 clade, almost no convergence within the M1 clade. So the M1 anoles in South America, almost no convergent evolution. Why that might be, that was a surprise. What that suggests is that the M1 anoles, when they evolved their morphological differences, they quickly spread out across all of northern South America. So for example, when you see a twig anole in northern Colombia and one in the Pacific side of the Andes, they are closely related, members of the same clade, as opposed to being convergent. That perhaps explains this lack of, because there is great variety throughout South America, it just hasn't evolved convergently. So in this case, I want to point out that it's the M2 and the Greater Antillean anoles that show the same pattern. Finally, we looked at ecomorphological relationships in several different ways. And this too is a bit of a complicated slide, and there certainly are differences between the two mainland anoles in green and in blue. My apologies, the colors are not well chosen for those with color vision issues. There are differences in the slopes. Nonetheless, the mainland anoles are much more similar to each other than they are to the island anoles in almost every case. And so in this case, it is the mainland anoles that ecomorphologically are more similar. So you add this all up, and basically what you find is that there's no single pattern of radiation, that each of the three groups has radiated in a different way. That is to say, the M1 group is more similar to M2 in some respects and more similar to island anoles in some respects. And that's true for all three clades. And so basically, although all three groups have radiated widely, each one has done it in its own special way. There's no one way to adaptively radiate in these anoles. And finally, I want to get to the last question in the last few minutes, and that is, what happens when adaptive radiations collide? We have these two mainland radiations, and as you will recall, they evolved in very different areas. The M1 clade was in South America, the M2 clade was in Central America, but then when the Isthmus of Panama began to emerge, the two clades finally came into contact. What happened? They were certainly very similar radiations. Well, there's two possible outcomes. On the one hand, they may be so similar that there's basically a standoff, what's sometimes called a priority effect, keeping each one had already filled up the niches. I know I use this thinking a lot that you might complain about, but they each filled more ecological space, and so they couldn't be invaded. That is documented in some places, even in some anoles elsewhere. The alternative is that they are able to coexist long enough, because there are some differences, that character displacement could lead to enhanced differences permitting coexistence. That certainly might have occurred as well. Well, what do the data say? And the short story is neither explanation fits the data perfectly. In fact, neither one fits them at all. It's something kind of in between. Well, first, let's look at the distribution of the clades. And here we see the most interesting phenomenon. The M1 clade, diversified in South America, the Isthmus of Panama arises, and it barely is able to penetrate into Central America. Only 9 species occur in lower Central America, getting up to southern Costa Rica. So they don't intrude into M2's ancestral land at all. By contrast, M2 has no problem moving into South America. It now occurs everywhere that M1 occurs, throughout the entire northern half of South America. About a third of the species of anoles in South America are M2 clade. So a very asymmetric outcome just in terms of biogeography. And remember, I pointed out that the M2 clade, despite being younger, has almost twice as many species as the M1 clade. That's part of this as well. So a very asymmetric invasion. Is there any evidence for character displacement? Well, there is a little bit of evidence for habitat partitioning, that when we compare the M2 anoles in allopatry versus the ones in sympatry with M1, we see that the M2 anoles in sympatry tend to perch lower on the ground. So there is a bit of an overall segregation, whereas the allopatric ones are in the middle. So that is habitat partitioning, which could set the stage for some sort of character displacement. However, when we look at morphological characters, we find no evidence of character displacement. For the most part, the M2 anoles in sympatry with M1 and in allopatry, sympatry, are essentially the same. And in fact, when there are differences, they are in the wrong direction. It is the allopatric anoles that are more different than the M1, than the sympatric ones. So there's really no overall evidence for character displacement affecting the M2 anoles. In fact, quite the contrary, the diversity of M2 in South America is equally as rich as it is in Central America. They seem to have just occupied the same ecomorphological space. Now, some of that is just due to migration of species into South America, but it's also due to some in situ evolution as well. So all of these lines of evidence basically suggest that the M2 anoles were unaffected by the presence of M1. They sweep through South America, they become very diverse, they occupy all kinds of ecological space. By contrast, as I've already mentioned, the South American anoles barely get into the north, and it turns out that the ones that do get into Central America are quite different. It is only the largest species of M1 that occur in lower Central America. You can see this shift in average body size. Even in South America, M1 anoles are slightly larger, and that difference is accentuated. So there seems to have been a filter that only allowing the very large anoles into Central America in the presence of M2. Well, we have no explanation for this difference. I don't know. There is one interesting parallel that those of you who are familiar with what's called the Great American Biotic Interchange, of the same thing happening with mammals when the Isthmus of Panama arose, it's somewhat similar in that the northern invaders invaded and diversified much more extensively than the mammals in South America moving north. Now, why that parallel might exist, I don't really know. And more generally, this is a topic that there's a lot of interesting research yet to be done. So to wrap up, mainland anoles are as diverse as island anoles. They've radiated in some different ways, and there are differences not only between islands and mainlands, but between the two mainland radiations. Predation is probably a more important factor affecting mainland radiation, whereas interspecific competition has probably played a bigger role on the islands, and that certainly is likely to be responsible for differences between the two areas. But most importantly, I want to leave you with the idea that there are many more questions than answers. There's a lot we don't know, and even this new study has opened up new questions about the asymmetry in radiation. And so there are plenty of research opportunities. Those of you graduate students